Thank you all for being here this afternoon. So yes, I'm Sarah Carey. I'm a PhD student at the University of Florida, and I'm going to be talking about some more sex chromosomes. Back to back, aren't you excited? Okay, so there's one big question that we've been working on in the lab that I'm in, which is Stuart McDaniel's lab, and it is, are there differences in molecular evolution between males and females? And so one obvious place to look for those differences is on sex chromosomes. And so these are the chromosomes where inheritance is correlated with sex, and they can contain a lot of loci. So you can see here in our gummy worm karyotype here, these are the sex chromosomes down here. And the systems folks are probably most familiar with are that of the XY and the ZW systems, but you should note that those are both diploid sex chromosome systems, where XX, my W migrated, but XX and ZW, they typically correlate with being female, and then the XY and ZZs typically correlate with being male. And because these are diploid, when they are sex expressing, they make their gametes by meiosis. So those gametes then unite, fertilization, back to being diploid again. So the important thing to remember for the sake of this talk is, in these systems, only the Y or the W are sex-specific, and only the Y or W have suppressed recombination. But there's another sex chromosome type out there. There's the UV sex chromosome system, and this occurs when sex expression happens in the haploid stage. And so in these species, the U correlates with females, the V then correlates with males. And importantly, because they are already haploid when they're sex expressing, they make their gametes by mitosis. And these gametes will unite, fertilization, and the only time that they are diploid is in this monomorphic, non-sexed individual here. And then meiosis, back to being haploid. So what's great about this system, that's in contrast to those diploid systems, is both the U and the V are sex-specific, and both the U and V have suppressed recombination. Okay, so in diploid systems, this lack of recombination can be confounded with sex-specific inheritance. So for example, the non-recombined Y chromosome in males. It can be confounded with sex-specific mutational processes or sexual selection. But in this UV system, we can start to tease these effects apart. And so the system that we are using to address these questions is that of Ceratodon purpureus. It is a moss, and it has UV sex chromosomes. So yeah, the U is the females and the V is the males. This species is sexually dimorphic, so you can see in this picture here, the females typically grow much larger than males. Males make these adorable little flagellated sperms, and then females make eggs, and they nurture the offspring. And so there's lots of potential for genetic conflict in these, in Ceratodon and other mosses. And what we're really, really excited about is we recently received a chromosome scale assembly for Ceratodon purpureus for both a male and a female isolate. And so these assemblies were actually done by the Joint Genome Institute. So we thank these folks here, Jeremy Schmitz, Jerry Jenkins, James Grimwood, Carrie Berry, and a ton of other folks for these really awesome assemblies we have now. So this is a high C contact map, and I don't want to belabor too much about what it all actually means. I just wanted you to see our chromosomes. So each one of these boxes represents one of the chromosomes. And we have 13 of them. It's about 360 megabase genome. But my absolute favorite part about this is that chromosome is the sex chromosome. So that giant one right there is the sex chromosome in Ceratodon purpureus. So to break that down, that is, the U and the V both are about 30% of the genome in total base pairs, and they're about 12% of the gene content. So we have almost 3,400 genes annotated to both of the sex chromosomes, and when we start doing blast type analyses, about 2,000 of them or so are homologous. But in addition to our genome, we have some other genomic data that we get to work with. So these are where our genome isolates are from, so our male and our female. But we also have 23 genome sequence isolates. So they span a nice latitudinal distribution here, and then one from over here in Australia. And then we also have 20 transcriptome isolates, which are from some same overlapping populations as our resequenced data. And so these are the data that we're going to be using to address this question. Are there differences in molecular evolution between males and females? just a reminder, we're talking about haploid sex chromosomes. So everything we're looking at here, both the U and the V are sex-specific and have suppressed recombination. And so there's some expected consequences for putting genes on sex chromosomes. Because of suppressed recombination, we should expect to find things like decreased codon bias, and I'll explain some of these things a little bit later. But because selection is not as efficient in regions of suppressed recombination, we expect codon bias. Because of sex-specific inheritance and suppressed recombination, we expect to find a lower effective population size. And because of sex-specific inheritance, these genes are a little freer to evolve or to generate, depending on how you want to look at it, which we're going to measure at something called TNDS. So what we're really going to do is for these three metrics ask, are they different between the sexes? All right, so codon bias. So if you're not familiar, this is the suite of optimal codons that are selected for translational efficiency. And so when these genes get put inside of regions of suppressed recombination, selection is less efficient, you start to see a little less of bias towards those more optimal codons. So what I did was I took our genome annotations, genes annotated to our sex chromosomes, and I filtered for 200 codons, and I threw out anything that didn't have one count of a gene expression on it. Just kind of get rid of some things that might not be real. And this is what we find. So things that are more biased are going to be down here on the graph, and less biased are higher up here, so using more codons. And we can see that the sex chromosomes are significantly higher than our autosomes. Although when you look at the relative number, it's one more codon that they're using. Significant, but one more codon. But the question we really want to answer is between the sexes, and no, there is no significant difference between the sexes in terms of codon bias. So this means that purifying selection is acting equivalently, at least in terms of this metric, between the sexes. Effective population size, though. So you can have differences in effective population size on sex chromosomes simply because of the number of them that exist relative to autosomes. So in the case of an XY system, the X chromosome has 3/4 of the effective population size at neutral equilibrium because there are 1, 2, 3 copies of it in a population relative to the 4 autosomes, which are in purple. And then the Y chromosome has about 1/4 at neutral equilibrium. But in UV systems, there's 1 copy of a U for every 2 autosomes and 1 copy of a V, so both of them should start off at 1/2. So easier math. And there's also a really nice other note that I want to drop, that with UV systems, we can actually use homologous loci to look at these metrics. Because with the XY system, the X's are much more similar in population numbers to autosomes, so they get compared to that, but the Y is so much lower that it typically gets compared to other parts of genomes, so like mitochondrial or chloroplast genomes. So here are the X's, we're actually comparing homologous genes. Okay, and so what we did was we mapped our resequence data that I mentioned before to our chromosome, or our genome, and we used nucleotide diversity here for our proxy for effective population size. And so you're going to notice that I have the actual absolute values of these sliding window analyses covered. I don't want you to pay attention necessarily to the exact number I'm showing. I want you to see the patterns, because they'll be really informative, and you'll see them here in a second. But just for the sake of clarity, also, this is an incredibly polymorphic species based off of a lot of other data we have. So we're still kind of trying to work through exactly which mapper we want to use. Mappers don't like SNPs. And which program we want to use to call SNPs, so we're working on that. But there's one pattern that comes through every time we do it, and that's what I'm going to show you here. So this is now the U sex chromosome. So compared to the diversity of the autosomal chromosome 1 here, U sex chromosomes typically have anywhere between 1/4 to 1/2 the level of nucleotide diversity of an autosome. When we look at the V sex chromosome, which is the male sex chromosome, it's anywhere from about 1/20th to 1/8 of the autosomal diversity. So they are much, much lower. And so this to us was really exciting, but then also not super surprising, because as I mentioned before, this is a sperm broadcasting species. So we suspect that reduced effective population size in males is due to sexual selection. That is, males have an increased variance in reproductive success than females. Okay. And finally, TNDS. So DNDS is the measure of the number of non-synonymous to synonymous changes in proteins. And so this is going to be our measure of evolution on proteins. And so what we did here is we took our annotated genomes that I've been talking about in 3 of our transcriptome lines, and all other existing MOS RNA-seq data, which I want to stop and thank these folks down here. Sana Olson, Sana Hutton, the folks at the 1000 Plant Transcriptome Project, and Matt Johnson and Warren Wickett. These folks either gave me access to data really early or have given me data that they haven't published and maybe aren't going to. So I just want to point out it's the generosity of folks like this that have made this project as far along as it is and made it such a delight to work with. So thanks. Okay, so we took all existing RNA-seq data, and I'm not going to go into super details here, but I'm happy to talk about any of the methods at any point, if anybody's interested. But in short, I assembled genes, I clustered orthogroups, built some trees, filtered them a bunch, and then I ran PAML on about 350 trees. So my filters got rid of a bunch of the trees that we talked about before, because I said there are 3,400 or so genes on these sex chromosomes, so we only have about 10% of them left for doing PAML, but it is what it is. Okay, so what we find is this DNDS, that our females and males are not significantly different from each other in terms of DNDS, which is interesting to us. But if we split DN and DS apart and we look at them separately, they are significantly different. So DN here, so our non-synonymous mutation rate, males are significantly higher than females, and in DS, males are also significantly higher than females, which to us suggests that the males are experiencing a higher mutation rate than females are. Which this kind of boggled our minds for a little bit. We had to rack our brains because, like I mentioned, these make gametes by mitosis. There's no germline here. So we had to think about this, and then it kind of came to us like, oh, but wait, they are making a ton more sperm than the females are. So it's kind of not terribly surprising that they might have a higher mutation rate than the females do. Okay, so are there differences in molecular evolution between males and females? Yes, but it depends on which metric you look at. So in terms of codon bias, no. So purifying selection seems to be acting equivalently on these haploid sex chromosomes. If you look at effective population size, yes, the males are lower, which is likely due to sexual selection. If we look at protein evolution or DNDS, no, there isn't a significant difference between the sexes, but if you tease those apart, interestingly, there is a difference, which is likely due to a higher mutation rate in males. Okay, and if you want more moss, you should talk to my lab mate. So Chetu actually presented this morning, so if you missed that, I'm sorry, but she is still here, so you should totally chat with her about her awesome work from Alaska. And then tomorrow, Leslie Kohler is going to be giving a talk in Tucson H at 1:30 p.m. on genetic conflict and an ancient interaction between moss and microarthropods. And with that, I'd like to thank all of my collaborators, all of my undergrad mentees that have helped on various different components of this project, all of my funding sources, and the folks at the cluster, the supercomputer at the University of Florida, because they are responsive. Like Christmas, they have loaded a new module on there for me, and really appreciate all of their efforts. So, and thank you.